Dear participants, hello and welcome to this webinar. This is the first webinar of a series of 3. We will be having presentations from a number of people from China, Japan, the Netherlands and New Zealand. You see here the list of presenters for this webinar today. And before we start, we would just like to ask you to rename yourselves and to display the 2-letter country code before your name. If you are participating on behalf of an observer organization, you can display the acronym of the organization. We remind you that no interpretation will be provided during this webinar. We would also like to ask you to keep your microphones on mute. At the end of the webinar, after all the presentations from the specialists, there will be a question and answer session. At that session, if you have questions, we would ask you to please raise your electronic hands, and we will invite you to unmute and you will be given the floor. The chat function will be monitored for questions, and please also be reminded that this webinar is recorded, not for the question and answer sessions. Those will not be recorded, but the presentations will be recorded and will be made available on the UPOV website. I will now give the floor to Leontino Tavera to give a brief introduction. Thank you.
Thank you, Jessica. Greetings, everyone. Cooperation is one of the key benefits of the UPOV system, and the environment in which the system exists is evolving quickly. This brings the opportunity for DUS examination. The short introduction to this webinar is that the PVP operating environment, not just the environment for characteristics, but the PVP ecosystem is evolving quickly, and there are opportunities for DUS examination to gain efficiency and to be expedited through cooperation. Cooperation is, as a key benefit from the UPOV system, is a matter UPOV members have been actively discussing since long in search for opportunities to increase international cooperation in DUS. And this means to increase taking over DUS test reports. One of the ways that this has materialized was the establishment of the EPVP DUS exchange module. So this is an online service provided by UPOV, where UPOV members can request DUS test reports, as well as make available the test reports they generated to facilitate exchanges and expedite granting titles where examination results, test results for examination already exist. Taking over test reports often comes across questions in relation to the environmental influence in the expression of characteristics, and it is often mentioned that this could prevent the exchange of test reports and taking over test reports. So providing information about practices in this area could possibly increase international cooperation. The matter has been discussed in UPOV since long. For example, for over a decade in the 2000s, UPOV members discussed a project to publish variety descriptions. Within this project, UPOV members were comparing how were the variety descriptions generated for the same variety in different territories. This project considered a range of crops, agricultural, fruits, ornamentals, and vegetables. The example here on screen is of variety Kerpil, which was described in different UPOV members. And what you see on screen are the expressions or the environmental influence in the expression of the characteristics. So there could be discussions on the intensity of the color you see on screen. Years later, UPOV members came up with documents TGP5 section 6, where UPOV standardizes the test reports for exchanges. That document states that the description of the characteristics generated at the time of examination are linked to the circumstances of DUS examination. This means that any given description will be linked to the conditions, to the place, to the date in which DUS examination was conducted. So, if environmental influence in the expression of characteristics is thoroughly considered in DUS guidance in UPOV documents and TGP documents, how do UPOV members organize bilateral cooperation? How do they agree conditions to exchange test reports in the understanding that anywhere a variety is grown, a description may be different or slightly different? So in this regard, you will hear from different UPOV members how DUS examination takes into account the environmental effects in the expression of characteristics, but moreover, how this does not mean that you have to repeat variety examination. More the contrary, why is it not necessary to repeat DUS examination at each UPOV member, thus and therefore taking full advantage of UPOV as a means to cooperate and exchange test reports? For today, we have 5 speakers.
Good afternoon and good evening. I'm so sorry for the misunderstanding, because in the afternoon I think it is finished, and in the evening maybe the meeting will be related to the variety collection. So I forgot I need to repeat my presentation. Sorry. Yeah. Good afternoon and good evening, distinguished experts. My name is Liu Yanfang, and my family name is Liu. I am the Deputy Director of Kunming Subcenter of Ministry of Agriculture, PR China. It is my honor to have this opportunity to report our research on the impact of environmental variation on color trees expression, and we also want to discuss the challenging to DUS testing and corresponding solution. Okay, and yeah, my presentation. Oh, okay. My presentation includes 2 parts. One, I want to show some cases, examples of environmental effects on color we have collected. And in the second part, I want to show how we conduct our test and our some consideration. I think many experts have countered these problems because many of you may already observed color variation under different environmental conditions. These 2 figures are from a paper which we have published in 2014. In this experiment, we used two varieties, one named as A1, variety A1, and the second is named as variety A2. And meanwhile, we designed three environmental conditions, which are B1, B2, and B3. In B1, it is in greenhouse, and in B2, it is under the shelter. And in B3, it is in an open field. So the difference among the three different environment conditions include the light intensity, because we can see in B1, the sunlight is weak and the temperature is high, and in B2, the sunlight is medium and temperature is also medium. And in B3, the sunlight is strong and the temperature is low. I need to mention that the three different environments are all in one location. So I think the difference may be little, but from figure 1, we can see it is a figure from the early observation stage. We can see that across the three different environmental conditions, there are clear difference in anthocyanin expression on the outer gloom. The preliminary conclusion is that the strong light and low temperature can enhance anthocyanin expression. And also in figure 2, we can see the difference between figure 1 and figure 2 is that the observation stage is different. Figure 1 is the early observation, and the figure 2 is the late observation. And we can see the figure 2 also shows similar variation trend. So maybe the influence are similar. And this is another cases which are connected from 2019 research in 2019. For we used partial buttercup variety, and we grow it under different conditions. But the sunlight between the two conditions are similar. The only difference is that the light conditions are similar, and the temperature drops by 5 degrees centigrade. And we can see from that both the upper sides and the lower sides of the petals, there shows significant color difference. And the color variation trend include lower brightness and stronger red intensity and weaker intensity. So we can see when the light is similar, and only 5 degrees centigrade temperature difference, and that will cause great difference on the color variation. And also here is some cases. These photos are provided by a company. They are under different temperature and sunlight. And the petal colors changes not only in, we can see not only in color intensity, but also between the single color, and we can see the left one is a single color and the right two are multicolor patterns. For example, so we can see there exists a clear difference. Because these data are provided by ornamental crop company, and the environmental factors difference are not fully known, so we don't know why there are so huge difference between these variety, because they are one variety, different plants. Okay, now we have conducted related research, which we will not give details here because the time is limited. And in general, we found that under different environments, certain transcription factors regulate main or branch pathways of plant pigment expression, which will cause color changes. So we can see this is a complex issue. Different crops have different pigment metabolic pathways and transcriptional regulation mechanism varies across environments. Therefore, returning to the testing itself, here are some key points we need to consider. Firstly, I think a uniform testing environment is critical. For example, we usually say that a side-by-side testing mode can be greatly reduce the environmental effects on color differences. So we think a uniform environment, including not only temperature and humidity, but also consistent and uniform light for both candidate varieties and their reference varieties. We need to pay attention to factors such as the lighting, the shading, and the angle of light exposure. All of these factors need to be considered. And the second one is caution. We need to pay attention to what we use, especially when we use the color characteristic for similar variety screening, especially the characteristic of color intensity. For example, when we use the intensity of unsanitary coloration, we had better not to use them to screen, use them as screening parameters. Instead, if the color characteristic is very important in screening the similar varieties, we can focus on color family and qualitative aspects. And thirdly, we also need to be careful when we use color trees in intellectual property disputes. For example, I once participated in infringement cases of PBR. And in that case, the crop is New Guinea impatient. And the lawyer pointed out that there are differences between the petal colors in the testing report and the actual plant colors. In my opinion, this approach has certain shortcomings. On one hand, color deviation can be introduced by imaging equipment as well as operating equipment. And on the other hand, color variation can also be introduced by environmental difference. Sorry. Okay. Also the side-by-side testing can minimize the environmental effects on color differences. We still need to record color measurements in database as the key variety description information. So given the environmental influences mentioned, we proposed describing color using an advanced way, such as the qualification description based on standard color system, as well as the qualitative description based on color family. Now, let's look at some problems with the current RHSCC method. Firstly, RHSCC contains around 900 colors. As we know, they are totally maybe 6 million colors in nature. So the coverage of RHSCC is only 0.015%. And this figure shows that the petal colors of ported carnation, and all of this research are published in 2022. which were mapped onto the, we can see the color of connection were mapped onto RHCC, and we found that many of the colors are not covered by RHCC. So that means when we give the results of RHCC to these varieties, and the results will not be trustable. And secondly, we also found there are maybe totally 6 colors or 30 pairs of colors in RHSCC, which can be visually distinguished in pairs. Their color difference are below 3 delta E, which means they can be separated by human naked eyes easily. This will lead to some problems. For example, the same color may be assigned to different RHSCC codes. So, because RHSCC is neither qualitative or quantitative, and meanwhile, color traits are easily affected by environment. So, it is difficult to study or summarize color changing patterns using RHCC codes alone. And therefore, we propose establishing qualitative and quantitative technical system for color analysis. This would not only make color testing more accurate and efficient, but also support future research. For example, the research on environmental effects on color difference and the mapping of molecular markers associated with color characteristic, and identifying the transcriptional factors that are characterize color expression states. And we have made some progress in quantitative and qualitative color analysis, developing the software that can use CIE-LAB for qualification, and the Momser color space for CIE-LB for quantification, and the Momser color space for qualification description. And we look forward to presenting this at technical working group meetings such as TWO or BMT. Yeah, thank you for your attention.
Good morning, good afternoon, everyone. I'm Yufeng from the Xinzhou Subcenter of Plant Variety Tests, Maharashtra, China. Today, I can show our recent finding titles evaluation of spatial temporal consistency-based DUS traits. Let's start with some background. As the world's second largest maize producer, China accounts for 43 million hectares and 23% of production. It's over 8,000 DUS applications every year, and causes a potential real backlog. So here's a big question: can we shorten the testing cycle without losing scientific rigor? And that's what we are focused on. Currently, the UPOV and the Chinese regulation requires maize DUS testing across at least 2 independent growing cycles. However, most tests still following the 1 location, 2-year model, which spans at least 24 months. To overcome the efficiency backlog, we tried out a 1-year 2-location framework. If feasible, this model could cut the testing duration down to just 12 months. So we conducted a multiple environmental trial. Let's take a look at the details of the experiments. We implemented a predominant block design across 2 geographically distinct testing sites in Shanxi province, Xinzhou 4044 to 4045 and Datong 4045. Following the China National DUS testing standard, we evaluated 170 major genotypes, including 21 inbred length and 196 hybrids, based on the 69 basic characters. Statistics analysis of CV, PCA, and APMA clustering was performed using SPSS and origin. As shown in table, two locations exhibit significant contrast across in the environmental variables, especially altitude, temperatures, precipitation, and sunshine duration, etc. These diverse conditions enable comprehensive evaluation of the traits across independent growing cycles. Now, let's turn to results. Using a 30% CV threshold, we identified 20 core stable traits. For example, take leaf blade width. It remained highly stable across all three environments, with CV values of 11, 9, and 12% respectively. In contrast, 16 traits were highly sensitive to both temporal and spatial variation. For example, the number of primary lateral breaches showed CV of over 60%. And 3 traits showed instability only temporarily, while none did so only spatially. As expected, hybrids and inbreds also differed markedly. For example, the ear length has a low spatial CV of just 8% in hybrids in 2025, but its temporal CV reached 38% in 2024. The spatial temporal divergence between the hybrids and the inbreds stems from the hybridization catalyst effect. Inbreds exhibit strong phenotypic canalization, and hybrids, on the other hand, are more sensitive. For example, plant heat. Just as we have observed the ear length, although a hybrid's ear length may be consistent across locals within a single year, annual weather function fluctuations such as rainfall or temperature can amplify the phenotypic noise. Then we use the PCA to reduce the phenotypic data examinationally, and the first 12 principal components explained nearly about 75% of spatial variance, while the first 11 explained about 68% of temporal variance. This indicates that the spatial environment plays a dominant role in shaping the overall phenotypic texture of maize. Finally, we distilled the 39 traits into 77 core traits. This includes plant and leaf dimensions, tassel and globe morphology, and including quantitative, and pseudo-quantitative characteristics. And collectively, these 7 traits capturing about 75% of intervallic variation. Next, based on the DUS traits, we used the APMA cluster analysis to assess classification stability across 106 genotypes. Especially, the clustering returns rate between Xinzhou and Datong in 2025 reached nearly 92%. Temporarily is nearly 95%. Both rates exceeded about 90%, confirming that multiple location trials may be as stable and scientifically valid as multiple years trials for DUS testing. A free cluster shapes was observed along with our earlier finding. Finally, we evaluated the consistency of DUS tests across 106 major genotypes. For indistinctness, the spatial model achieves nearly 94% consistency rate, nobly higher than the 87% seen in the temporal model. For uniformity, the spatial model also outperformed research about 96% with versus 93%. When we look at the final over qualification rates, the two models were much closer. And it's 83% for spatial and 87% for temporal. Our data from clear spot for transforming to a 1 year to location trials is a feasible and effective strategy for maize DUS testing. In conclusion, our results demonstrate that the 1 year to location model can accelerate maize DUS testing, addressing China's current efficiency bottleneck and the regulatory background. Future, we will expand to more ecological zones and use multiple data to multiple G/E interactions. So, and the DNA markers and the phenotypic observation will be integrated, and this further enable a more precise DUS testing system and better meeting the testing demands. That's all for today. Thank you.
Okay. Sorry. So I am Yoshiyuki Ohno from Plant Variety Protection Office, Ministry of Agriculture, Forestry, and Fisheries of Japan. So I would like to introduce environmental effect in expression of characteristic in DUS examinations. So our experience for soybeans. Today, we will have the three main topics. The first one is breeding activities in Japan. Second one is environmental impact on soybean in Japan. And third one is experience DUS examination on soybean. So, so here we have breeding activities in Japan. So breeding objectives, vicissitude. We have the table. So the past breeding scope. So I can say the breeding objective was improving yield, stable production, and soil transmitted disease resistance. So past scope was no relation to the any gene locus. And also past breeding method was hybridization breeding, mass selection, and pedigree selection. So on the other hand, current 15 years scope pot dehiscence and disease resistance, mosaic virus, insect resistance, for example, cystonematode, severe climate tolerance, tolerance for the cadmium, dominant gene for pot color, dominant gene for flowering time. So these 15-year scope is relation to the some DNA markers. So our current breeding method is pinpoint breeding by using molecular marker and backcrossing, and also gene genome editing, which is to speed up breeding, and reduce the time and cost. So Japan has demand of the new plant variety for agriculture production is increased, and plant breeding technology is improved to address such demand rapidly. So here we have the general information on climate of Japan. We have 4 distinct seasons with climate ranging from Arctic in the north to subtropical in south. Different climate conditions between Pacific Ocean side and Sea of Japan side. Northern area, warm summers and very cold winters with heavy snow on the Sea of Japan side. And also eastern area, hot and humid summers and cold winters with very heavy snow on the Sea of Japan side and in mountainous areas. And also western area, very hot and humid summers with temperature sometimes reaching 35 degrees Celsius or above, and moderate cold winters. Okinawa and Amami area as subtropical oceanic climate. These areas have hot and humid summers with temperature rarely reaching 35 degrees Celsius or above, and mild winters. So here we have the environment impact on soybean in Japan. So I already mentioned the elongate geography from north to south, and soybeans growing area is different in elevation. Each varieties are significantly different relative to the production area. The favorable temperature for the growing is 25 to 30 degrees Celsius. A minimum accumulated temperature is 2000 degrees Celsius. The average temperature more than 12 degrees Celsius at least 120 days. Detailed regional zones are established to select the most suitable varieties. And the critical day length is generally 12 to 14 hours. If the short day conditions are less than 12 to 14 hours, flowering is progress. So temperature sensitivity and light sensitivity are varied depending on the variety and regional zone. And most important point to be selected varieties according to the northern latitude, day length, and temperature. So here we have the categories into the 3 ecotypes of soybean to adapt several climates in Japan. First type, summer soybean type. The feature is sensitive to the temperature. Insensitivity, short day reaction for high northern latitude area. So summer soybean type has a risk of the growing unsuitable area. It is high temperature causes to too early flowering with major plants. And also Japanese soybean has intermediate type, second color. This type is intermediate between the summer and autumn type. And third one is autumn soybean type. The feature is sensitive to short day reaction for low northern latitude area. Risk is long day condition caused too late flowering with overgrowth plants. So here we have the map of the Japan. So breeding and agricultural production area is detailed and divided from zone 0 to zone 7. So the production area has very detailed zone. For example, summer soybean type here, and intermediate type here, and autumn soybean type here. So our experience, DUS examination on soybean, to optimize DUS examination on soybean, it was considered to set a regional set of example varieties for each growing zone. Japanese national test guideline divided by six zones in national land with regional example varieties. So you can see the here, Yufeng is North cold region as plant height is 5 medium example variety, but the different region, cool climate region, the Yufeng is little bit shorter plant height. So here we have the simplified images for the plant height on the soybean from high northern latitude to the low northern latitude. So it becomes some long length to the low northern latitude. So finally, I can say the traditionally important soybean food products in Japan. Here we have some processed food from the soybean. Thank you very much for your attention.
Good. So, good morning, good afternoon, all of you. I'm very glad to have the opportunity to tell something about this important topic, I must say, because it's really important in the US testing. So this is what my presentation is about. I'll tell something about the background, which has been told also already. What is environmental effects? How to avoid it, because that's an important thing. And I give 4 examples of cases in our crop species where we have a kind of solution to work in these crops to avoid this effects, let's say. Now here you see the same thing as Leontino already explained. There was the Petunia study where you see differences in color. The same thing was also done by Boulineau for UPOV in 2012 for peas, and there we saw the same effect. We saw that even qualitative characteristics were mostly identical notes if you compare different descriptions from several countries, but even for those qualitative characteristics, 1 to 8% of characteristics were not the same note over the different characteristics for different countries. Quantitative characteristics, now that you can understand, that's much more complicated. For example, we saw that in 80% of the characteristics were the number of nodes between the United Kingdom descriptions and the France description differed in 1 or 2 nodes. So that was a drastically difference, let's say. Also, what was explained already, the RHS color codes, of course, they can be very different. Sorry. So what is very important is to realize that using pictures is not always that clear. So anyway, you can see environment effects play a very important role in differences in variety descriptions and in differences in pictures. But environmental effects are, of course, not the only case or the only problem. Like in qualitative characteristics, you can imagine that this 1 to 8% of differences in descriptions are not only differences because of environment. Probably there are also mistakes at stake. So here to explain what you see here, and that was explained also by Yoshiyuki, you see here what you expect normally that we have environmental effects. The expectation, however, is that if you go to another environment, the scale remains the same. So the expression of characteristics goes up or down, but the differences between varieties have the same. So we think use of example varieties normally solve the problem. And that's in many cases is the case, let's say, but reality is that it's not always true. And that was shown by Mr. Yoshiyuki. You saw example varieties also change in environment. That's because of the environmental times variety interaction. So if you go to another environment, and let's say comparing those varieties expression, they are not in the same direction. So in another environment, you see variety F goes down, variety B goes up. So that's why it was a good solution for in Japan to assign regional example varieties. What we do as much as possible, because we have in the Netherlands, we have a small country, we have mostly one place of examination here in Roelevarensveen in the west of the Netherlands. We try to avoid environmental effects as much as possible. And I will show you how we do it in showing some examples. Another case which is important to tell in which, let's say, in which steps of the DUS process are those environmental effects important. And then I see at least 2 important things. One is when we are talking about distinctness, where we are looking for clear but especially consistent differences. And we run into problems, let's say, when we see environmental effects. So differences can, in 1 year, another season, can be different than in another year, another season. And then we have problem into detecting consistent differences, let's say. So that is one thing we realize and you try to avoid by, for example, testing in just 1 season. Another thing which has been touched already also is exchange of reports, and that has to do with, let's say, all the nodes in the variety descriptions. So when we have a variety collection, we put all the data in our database, and we do that by collecting, let's say, all variety descriptions from different countries from ourselves. So it's very important to make harmonized, comparable descriptions. But environmental effects cause that those descriptions are not always comparable, let's say. And that's I can show you what we have for kind of solution for that. So 2 important steps where it's important to realize how to avoid those environmental effects. I come to the first example, where we have in ornamentals, we have agave hybrids. We call them sometimes mangave. where we have crosses between agave and Manfreda. And because we have crosses with Manfreda, especially in the Manfreda species, there is a lot more anthocyanin. So what we see, that you see differences. It's good to realize in this species we have only one growing cycle. So we don't have to do with, let's say, to compare results from one trial to the other, because we have only one trial, we have only one growing season, but we make pictures. So it's important to realize. And what we see is that normally this species is grown outdoor. It's possible, you can grow it, but in the Netherlands, winters are too cold, so it's very risky, let's say, to grow it outside. So we have to grow it indoor, and that also makes that we have more stable conditions. But what we see, if we grow inside and outside, we have done a test. What you see here is that we have differences in expression of especially anthocyanin, coloration, and attitude. And what you see here, these are pairs of pictures, let's say, where you see that the same variety is grown inside and outside. And this makes you realize that it's really important to test, let's say, each variety under the same conditions. I mean, if you do it 1 year outside and 1 year inside, then you see the differences here. For example, here you see the same variety under outside conditions. It's much more red, much more anthocyanin than under indoor conditions. Even the attitude, you see this is the same variety. You see there is a big difference, and it's just caused by the environment. So it's very important that you realize that there is such big influence of environment on the expression of some characteristics. And how to avoid? We say, okay, it's what we do. We use the same conditions for each season. So we grow always inside. We never grow inside and outside, because then the results are not comparable. You don't have comparable descriptions. You don't have comparable pictures, because they will be very different. Also, this picture shows that there is a big difference from outside growing and inside growing of the same variety. So the solution, partly solution for this, is that you use the example varieties, but I explained already that use of example varieties does not solve completely the problem of these differences in description. If you have different environments, you see also pictures will not be comparable. So to solve this, we make use of only one type of growing. So we grow it always inside. So we have comparable descriptions, we have comparable pictures, and from each variety, let's say what we receive, which is not has not been let's say tested for the US in the Netherlands, we have a description, but we make our own in-house description from the new varieties, which we include as similar varieties or example varieties in our trial. And this enables us to have, let's say, comparable descriptions in our database for the use of treating applications in our DUS process. So this is an example for agave. Next example is for pepper, where we have indeed 2 different growing cycles, and which makes it more complicated. We have different sowing times. We have 3 different sowing times and trials in the Netherlands. So what we see is that we have a spring trial and an autumn trial. There is a huge influence of the amount of light and the amount of temperature, which causes differences sometimes in the expression of, for example, intensity of color. So some varieties are in summer, in spring trial, are much darker red than in autumn. Other varieties have the same expression of this intensity of red or yellow coloration in spring and in autumn. Also, the same effect you see in fruit size. Some examples are very stable in expression. Some varieties are less stable, and maybe in spring they have bigger fruits, and in autumn they have less big fruits, because they are much more affected by the fact that in autumn there is less light and the temperature are lower. Also, anthocyanic coloration of the stem is influenced in this way, that you see an uneven expression between varieties in autumn and in spring. So what we see is that descriptions are only partly harmonized by use of example varieties, because also example varieties can have a different expression, and pictures will not be comparable if we grow them in spring and in autumn for the same varieties. So our solution is that the same types, the same groups of varieties, we grow in the same season. So if we have a group of varieties with a certain resistance type, which all belong and have to be compared with each other, we always grow them, for example, in summer. Other types we always grow in autumn. In that case, we can avoid those environmental effects where you see influences that in summer there will be different behavior than in spring. The other thing is that by using let's say the same growing season, we avoid that we end up with the difficulty that we cannot compare the results of the two growing cycles, because we have two growing cycles for pepper. We have to be able to draw conclusions on consistent differences. And if we grow an application the first year in autumn and the second year in spring, then there might be difficulties to see consistent differences, because in one trial they will be lighter red fruits, and in the other season there may be darker red fruits. So to avoid this, we also grow the same type, the same groups we grow at the same season. So we have less difficulties in drawing conclusions on consistent differences. So we have a solution for comparable descriptions, we have a solution for comparable pictures, and we have a solution if we have, let's say, received descriptions and varieties from outside the Netherlands, which have been tested outside the Netherlands. We always make our own in-house description to make descriptions comparable in our database. Otherwise, if we put, let's say, the foreign description in our database, there are a lot of examples of characteristics where the nodes, let's say the expressions of characteristics, will not be comparable. So from each variety, we always make our own in-house description. The third example is cauliflower, where we also have two different growing cycles. We have even 4 different sowing times and trials. In cauliflower, there is a special thing where over different locations and countries, especially the earliness is very difficult to compare. If you look, for example, you have examples where, for example, varieties have an expression in the Netherlands node 4, so that's early to medium. In France, they are medium to late, and that's for the same varieties. Also, over seasons, you can even see bigger differences. We have sometimes types, the Erfurter snowball types, where if we grow them in summer in the Netherlands, they are early. If we grow them in autumn, they are late. I must say it's quite exceptional, but we see this kind of things. So what we see, and this is especially the case for this characteristics in cauliflower, that we have no example varieties in the UPOV technical guideline. You see here. You see the test guideline doesn't show here example varieties for the characteristics earliness. And this has to do with this fact that there is so much influence on environment that you cannot tell for every state, okay, this is a good example variety for early or for medium or for late or for very late. So in every case, we have to look for your own example varieties in your own trial, in your own circumstances, in your own season, because that can be different. So descriptions will be difficult to harmonize by use of example varieties, especially for this earliness, and pictures in this case are not so important. But what we see, we have two growing cycles for cauliflower. So let's say the first thing is that we have to draw consistent differences. So what we do, we grow our application always in the same one of the same 4 trials. Let's say we never do the same the first year in the spring trial and the second year in autumn trial, because then we can end up with incomparable differences. So we cannot draw conclusions on consistent differences, especially for earliness. So we grow the same groups and types in the same season, so we have comparable descriptions, and again we make our own in-house description for foreign varieties, which in the case of earliness in cauliflower is very important. I showed you sometimes there are very big differences between countries, so also between descriptions there can be very big differences. So it's very important to realize we really have to be very careful by including and by deleting, let's say, similar varieties from the trial on beforehand. So we grow everything, we make our own in-house description, and that is the let's say the note which we take into account when we search for similar varieties in our database. Very important in cauliflower. The last one, which is a little bit an odd example, but for us it's very important because we do a lot of testing for vegetables where resistances, claims for resistances are very important. And there we see even much more environmental effects, I would say, because we see differences between tests, but we see differences between seasons, which are caused by not only the varieties, but they're also caused by the disease. Especially fusarium disease resistances are very, let's say, very important to realize that the expression of the disease is not always stable over different seasons and over different tests. And we call this incomplete penetrance, which means that the expression of the resistance is not always the same. Our solution for these resistances is that we include, let's say, controls. Controls for validation and controls for calibration. So we know that, let's say, there can be a shift for resistant controls, for example, varieties in our trials. In one trial, resistant control can show a little bit more resistant reaction, let's say, than in another season. Here you see, for example, here you see a resistant variety which shows quite resistant plants, but you see also some smaller plants. You should think, okay, maybe these are susceptible plants, but it's not caused by the fact that those plants are not resistant. If you test, you can see they all have the resistance gene, but it's caused by the fact that you have incomplete penetrance, which causes that sometimes the resistance is not expressed well in some plants, in some seasons, in some tests. So this is really why it's important to have those calibration controls. Another solution is that you do not test in all seasons. For some diseases, you know, okay, we don't test in winter because then the expression is, let's say, less comparable with the other seasons. So including controls and testing in the same season is the solution for this environmental effects in resistances. Then, which is important, because Leontino told already, okay, we have a problem when we have exchange of reports. So when we exchange reports, we need those reports for inclusion in our database. We need those for searching for similar varieties. The solution is that, I mean, you cannot avoid the differences in in these differences in descriptions. But what we do, we always make our own in-house description of the varieties which have been described in other examination offices, to assure that we take into account the expression which is shown, let's say, under our circumstances. And even here in Europe, it's an obligation from the CPVO to do this, not include the descriptions as such from other examination offices, but you have to make your own description. So this is the solution when we talk about exchange of reports and working with a database with descriptions. So this is some aspects, I would say, about this topic. And thank you very much for your attention.
Good morning. Thank you for the opportunity to participate in this webinar. As with the other presenters, we're discussing our perspective on environmental effects and their relationship to DUS testing and examination. So, yep, as Carolyn said, my name is Scott Gregan. I'm a senior examiner with the New Zealand Plant Variety Rights Office. So I have a very brief introduction just to provide some context about how we operate as an authority with our examinations before I give some examples of our considerations around environmental effects. So this photo represents the entirety of our office. We're a small team where we consist of a technical lead and 4 examiners. And in terms of scale, depending on the year, we average between 90 to 120 applications per year. And this is across all plant genre and species. And then this also includes some fungal species. So around, we have around sort of 300 to 350 varieties under test at any one time being managed by our 4 examiners. So in terms of examinations, as I said, we're a small office. We don't centrally test and or produce variety descriptions for all our applications we receive and utilise a number of different testing arrangements that we manage on a species by species basis. In saying that though, we do run some centralised trials, and examples of this pasture species, grasses, clovers, fodder crops, etc., cereals and potatoes. And other testing arrangements, a number of our examinations are managed using cultivar centres or variety collections, where we grow perennial varieties, initially for examination, but then they are kept in the collections to use later as comparison and example varieties. And as I also mentioned earlier, we don't generate local variety descriptions for all of our applications. So we do take advantage of testing performed overseas, taking over test reports from other countries. And again, depending on the season, this might be between anywhere around 20 to 25% of our applications we would use an overseas test report for. And lastly, we also use breeder testing and testing on applicants' property for varieties where we don't, where our office doesn't have the resources to manage the growing trials, or where specialised growing conditions might be required, for example. But even with these last 2 testing arrangements, the examination of characteristics and development of the variety description is still managed by our office. So with that in mind, a little bit of background and moving on to the point at hand, our main focus really is that we build up our knowledge about how varieties, excuse me, about how varieties behave in different environments for DUS examinations and trials. We certainly don't try to control these differences. That would be hard to do anyway. And we don't exclude characteristics purely because of observed variation. And in fact, learning from experience is more useful for us to try to understand how environmental effects will influence our examination. And our focus shifts with the examination method or testing arrangement. only subtly really, but for centralised growing trials, our considerations are around knowing how comparator / example varieties will behave in trials for harmonisation. This of course comes with increased experience growing the varieties year on year. This is also important as many of our centralised trials are for open pollinated varieties where there is an inherent level of variation also. Developing national example varieties is important for us in New Zealand. Down in the corner of the world, we don't always have access to varieties listed as examples in test guidelines, or more importantly, in the context of this presentation, sometimes the scores of characteristics of example varieties grown in New Zealand can deviate from the test guidelines, and we have to take that into account for our examinations and our comparisons to other varieties and comparisons or our standardization to example varieties, for example. And of course, being diligent, we need to be diligent when we have known environmental effects with characteristics that are being used to define distinctness. For perennial species, the considerations really are much the same, especially with characteristics that are being used to define distinctness. Knowledge about the environment versus expression of characteristics can help us interpret variety descriptions and potentially help explain differences from other environments. For example, when we're using overseas test reports for our examinations. In addition, growing the variety and knowing what the plant material looks like when grown locally is also important for us. So even when we're using overseas test reports for examination, this often is not in isolation. We still like to, where we can, grow the variety for verification and comparison to the test report description and noting deviations and potential environmental effects. And this is an important point. This is not to say that we consider such environmental effects to be a problem or we are particularly overcautious. It certainly doesn't put us off taking over test reports and overseas examinations. But for our purposes, it is important that we are aware of these effects and we manage this through experience. And this, as the last talk was pointing out, this ultimately helps us manage and maintain accuracy of local variety databases and to accurately define VCKs for future grouping and trials. So that's sort of a brief overview about how, sort of on a sort of a broad basis, how we manage environmental effects with regards to DUS testing examination. So with the remaining time, I'd like to just give a few examples we have observed of our testing in New Zealand. So two areas where I'd like to say two main areas, but these are sort of two areas where it's common to see environmental effects on the expression of characteristics to do with the light environment and temperature effects, and potentially an effect of the combination of them both. I don't want to go into any mechanisms because these aren't quantified. We don't really know, but these are just some observations we have noted and are consistent observations through our years of testing in New Zealand. So New Zealand has a unique light environment and strong solar radiation, and we have observed differences with fruit skin colour, for example, and anthocyanin production in young vegetative structures, and these are potentially due to these environmental effects. And I'd also like to show some examples of temperature, which has observed effects on vernalisation, winter growth, dormancy and sowing time for our growing trials in New Zealand. So in terms of fruit skin colour, intensity of overcolour has been important for distinctness in New Zealand. We have had examples of this that have been important for determining distinctness versus other varieties. We had an apple variety, for example, that was very similar to its comparator, and its colour characteristics made it distinct in New Zealand, but when tested overseas, the increased intensity of colour was not observed. There does also seem to be a temperature effect with regards to colour intensity in apples. We've noticed this through ongoing testing and experience with year-on-year collection of data. Overall, low colour levels in apples have been noted in warmer seasons where we have warmer nights and lower diurnal variation in ambient temperatures compared to other seasons. Also, sticking to the colour and anthocyanin theme, in some raspberry varieties, we have noted less anthocyanin expression in young vegetative structures in New Zealand growing trials when compared to overseas variety descriptions. These were noted where we used where we took over overseas test reports, but we were also growing the varieties in our variety collections in New Zealand. And comparing them back to the overseas description. But even though we saw less anthocyanin early on in these sort of young shoots and vegetative structures, interestingly, the anthocyanin production was equivalent in later development in fruit, flowers, and dormant canes. So in this instance, comparing the similarities and deviations from the overseas description really demonstrated the importance of having plant material in the ground in New Zealand and being able to sort of quantify differences to the test report we use for the examination and build up that body of knowledge and that experience. We have a moderately temperate climate in New Zealand. To highlight this, our centralised pasture trials are autumn sown in New Zealand. They are sown in May, which is the end of our autumn, grow through winter, and are examined in early springtime. Consequently, with vernalisation, in quote marks, characteristics are important in our examinations. This photo was taken just at the end of winter, coming into spring, which show how much growth the plants had put on even over winter in our pasture trials. Additionally, in our pasture trials, we get significant variation in ranges of flowering time and heading dates between early and late varieties of pasture crops, depending on the season. Move away from pastures. More recently, we have used overseas test reports to examine onion varieties in New Zealand. A number of these varieties from their descriptions are spring sown from the European test reports, but these same varieties are actually considered autumn sown varieties in New Zealand. And generally, most onion varieties grown through the winter in New Zealand are considered spring sown, and this gets more pronounced the further north you go in the country, especially getting up to the top of the North Island, which has a much more subtropical type climate and very mild winters. So dormancy is another area we have observed environmental effects on DUS characteristics. An example here in this case was with an overseas blackberry variety. This variety flowered and produced fruit continually throughout the year, even in the winter, and even in the winter never seems to go completely dormant. These flowers make very early fruit, and the ripening of which overlaps with the flowering fruiting of the previous season. And so this makes the delineation of what's a primer cane and what's a floricane and where is dormancy, can make it a little bit confusing and provided us with some difficulty. But and this picture was actually taken in the middle of winter. The plant was still looking very leafy, continue to produce flowers right through the winter, throughout the entire season. So that's sort of an example where potentially more mild winters have an effect on, such characteristics such as dormancy. So thank you. That is the end of my presentation.